Hi everyone, welcome to this week's PGSS talk by Cherry Tsao titled Are You Listening to Your Gut? A Study on the Stomach-Brain Synchronization. Cherry comes from the Laboratory of Integrated Brain Imaging, which is a part of the Department of Biomedical Engineering and the Department of Electrical Engineering and Computer Science at the University of Michigan. The lab aims to accelerate neuroscience and artificial intelligence by developing novel tools to integrate neural imaging, recording, stimulation, and modeling. As a part of the lab, Cherry's research interests include neuromodulation, neuroimaging, and electrophysiology for basic and translational neuroscience. She aims to integrate different techniques to answer important medical questions, and her current research focuses on assessing the physiological role of the stomach-brain interaction. So, please give your attention to Cherry and her presentation.
Thanks for the introduction. I'm Cherry, and I come from LibLab. I'm professor at Stromanville. And in this presentation, I'm going to talk about a new study about the stomach-brain synchronization. So as a human, we are very familiar with our digesting, our GI. So we eat and we digest, we absorb nutrients. It's because it's essential. As a baby, when the baby is born, the first thing she do is breathing. The second thing she do is trying to find the milk. So it's quite essential. It's not only essential, it's very old. So in the evolution history, the first recognized the digestion system, it's come from the sea sponge, and it's already appeared on Earth millions of years ago. The digestion system is quite simple. It's just the food cells. It absorbs the water and find the nutrients in the water and pull the water into the tube of the sponge and then push the water out. That's the earliest and simplest digestion system. After millions of years of evolution, now as a human, we have a really complex digestion system, including the esophagus, including the stomach, and including the intestine. So it's including many parts of the tract, and also there has complex neuron system in the digestion system. So it can function independently even without our notice. We don't need to control, we don't need to tell our stomach to contract to push the food, and we don't need to tell the intestine to absorb the nutrition. So it's fully independent. Although it's independent in nature, but it also has close connection with our brain. It's not only have a hormone communication with the brain, but also neuronal communication with the brain. There is a lot of neural connections between the two. For example, for the esophagus, it's already afferent pathway connect to the brainstem, and for the stomach, there's not only afferent connection to the stomach, there is also direct connection to the stomach motor neurons from the brain, and so for the intestine. So in our study, we are going to focus on the major part of the one of the major part in the intestine tract, which is the stomach. So the stomach has a lot of influence on the brain. So based on some studies about some patients with the digestion disorder, they found that their brain activity is highly impacted by the disease. So it's the impact includes in the sensory motor network, which can control our sensory feelings, our motor functions, and it also affects a lot of high-level networks. For example, it can affect the network of Stelling's network, executive network, default mode network, and the emotional arousal network. It can affect how we pay attention to our study, and it can affect how we feel our study, can affect everything, every single detail. So it's important, but is there any problem medical problem related with the stomach-brain interaction? Yes. For many gastric dysfunction, for example, the gastroparesis, the functional dyspepsia, and some unknown nausea and vomiting. Some of them may have the disorder because of their gastric function, their stomach did not function well. But there is also a large proportion of the patients that they have healthy GI tract but still have the gastric disorders. Why? Because they either has the nerve damage that which connecting the stomach and the brain, or they have some central disorders. For example, a lot of patients have Parkinson's disease may end up with a gastric dysfunction, and some patients may have migraine. They also end up with a gastroparesis or other disease. So there is the disruptor of the stomach-brain interaction may end up with a lot of gastric disorders. And the gastric disorder will also affect how our brain works. In many studies, they try to find out whether there did a lot of study on the patient with gastric disorder, and they found that a lot of them have a brain disorder such as anxiety and depression, and the percentage is much higher than the normal population. And a lot of patients with gastric disorder often end up with some brain disorder, has higher chance to have a depression or anxiety. There are also it's not only medical problems, but also other things. So I don't know whether you have experienced that when you undergo high stress, you have high intention to eat. So there is some stress-induced obesity. Also, there is motion sickness. When you travel, there's nothing affecting your stomach, but after the traveling, you end up with nausea and vomiting and sickness. And instead of that, I believe most of us have the feeling that, oh, I have a butterfly in my stomach. Oh, my stomach is crunched. So it's may induced by because you are super excited or you are super stressful. So the stomach-brain interaction affects not only on some medical problems, but also affect us every day. So by studying the stomach-brain interaction, how could we benefit from this study? There is two main effects from the interaction study. First, it can help us to with the diagnosis of the gastric disorder. So take the gastroparesis as an example. So the pathology of the gastroparesis including three parts. One is that the gastric nervous system has some neuropathy occurred. The second part is there is some mechanical disorders in the GI tract. And the third major cause is that their autonomic system has some disorders. However, when in clinic, when the doctor tries to diagnose what causes the gastroparesis, there is the only way they can say whether it's caused by the autonomic disorder is by asking the patient, do you have headache? Do you have experienced epilepsy? And use such kind of way to see whether the gastroparesis is caused by the neural disorders. So by studying the stomach-brain interaction, we can help with this diagnostic process and identify the patient that has a disease caused by the neuronal disorder. Another aspect of this gut-brain interaction study is help us with the therapy of the gastroparesis. Right now in clinics, there is a widely used way to help with the gastroparesis is gastric electrical activity, or electrical stimulation. So they implant the electrode on the stomach and they electrically stimulate the stomach. By doing so, some patients have relief of the nausea and vomiting, especially in those patients has a gastroparesis induced by diabetes. But it doesn't work well for other type of the gastroparesis. And recently there is some appealing new therapy, which is the vagus nerve stimulation. by stimulating the vagus nerve, some patients have improved the symptoms of the gastroparesis. And in some other study, also use the vagus nerve stimulation. They also found that some stimulation parameter can help with the obesity patients. By stimulating the vagus nerve, their appetite was reduced, and it helped with the weight control. And the most advantage part of this vagus nerve stimulation is that it can be non-invasive. We can stimulate the vagus nerve through the device that outside our body. There's no surgical procedure needed. So, but the mechanism by of the vagus nerve stimulation is not well understood. So currently, it's just a trial and error. Some patients undergo the vagus nerve stimulation and their symptoms get improved, but some are not. So it's highly dependent on how we stimulate and what parameter we use to stimulate. And the different patients may tolerate the stimulation differently. So by studying the stomach-brain interaction, it gave us a tool to evaluate how is the stimulation effect just by simply imaging or recording instead of trial and error let the patient suffer. So what's a current understanding of the stomach-brain interaction? So people have investigated this problem using some stomach stimulation techniques. So it's a small surgical procedure that puts a balloon into the stomach, and we can blow water into the balloon. When the balloon inflates in the stomach, it can create pressure to the stomach wall. So by doing so, we can stimulate the stomach. So during the experiment, the water can flow into the balloon and flow out of the balloon, such that the pressure to the stomach wall can fluctuate over time, just like what is shown here. It's form a block design like stimulation to the stomach. And by correlating this stimulation paradigm with the neuroactivity in the brain, they can find the evoked gastric network. This gastric network highly involves the insula and is highly overlapped with the network that processing the pain sensation. Why does that happen? Does our gastric network is highly overlapped with the pain processing? There is a question mark here. One problem is that this stomach stimulation is artificial and unpleasant, and it's usually the stomach stimulation can induce the unpleasant feeling and the pain. So it's not strange that the gastric network found by such kind of stimulation is highly overlapped with the pain sensation. So there is some other ways to study the stomach-brain interaction, which is use of stimulation. So as described previously, the gastric electrical stimulation is used in clinic. It can also be used in the research study. So we can implant a small patch electrode on the stomach wall, and by as shown on the left side. By stimulating the stomach, we can see how the brain responds to this stimulation. But similarly, this stimulation is artificial, it's unnatural. So people rarely experience such kind of electrical activity to the stomach. Oh, excuse me. Yes, something went wrong. Sorry. So the stimulation to the stomach is only a small part of the stomach function, including the electrical stimulation and the pressure sensation. So the major part of the stomach is contraction. So as shown on the left image, it's an MRI image that captures the stomach dynamics. And the right circle show the contour of the stomach, and including the fundus, the corpus, which helps to make the food, and the antrum, which push the food into the intestine, and the duodenum. This is the first part of the small intestine. So the stomach is always contracting. That's a major function of the stomach. It can help to make the food and push the food into the intestine. So our question is, does our brain listening to the stomach contraction, which is it's a normal stage of the stomach? The answer is probably yes. There is a recent study support this hypothesis. So what they do is that they record the gastric electrical activity in humans and try to find whether the electrical activity is synchronized with the brain activity. And they found that the gastric activity is fixed locked to the brain activity and illustrate intrinsic gastric network. So far, that's the only evidence suggesting that there is a brain gastric network that's receiving the stomach input all the time. So in our study, we want to validate this stomach brain synchronization. And in the next step, we would like to find the origin of the stomach brain synchronization, whether it's neuronal and which nerve is contribute to the synchronization. Lastly, we would like to uncover some other format of this synchronization, because the stomach not only have the periodic contraction, the contraction can also change. The strength of the contraction can also change over time. So our goal is to uncover other format of this synchronization. First, how do we assess the stomach activity? As shown previously, imaged by the MR machine, so we can capture the stomach dynamics, which is the contraction. So the stomach contracts all the time, but where does this contraction come from? It should start from the structure of the stomach. So the stomach have a few layers. The most inner layer is the mucous layer, which responsible for some chemical sensing and the stomach acid secretion. On top of that layer is a muscular layer. There is two muscular layers in the stomach. One is a circular muscle, which is circle around the stomach. Another is longitudinal muscle, which align with the stomach structure in this way. And in those muscular layer, there is some gastric neurons embedded in the layers, so which is called the ICC neurons. And these ICC neurons cluster together to form a small clusters that cover the whole stomach. And for each cluster, they can generate some electrical activity, and the electrical activity can propagate and affect other ICC neurons. And the electrical activity can affect the muscle and make the muscle contract. So that's how the stomach contraction functions in our humans. Then, so how should we recall this stomach activity and compare that with the brain activity? So we choose to use the electrical recording instead of the morphology recording. So the reason is, first, it's the electrical recording can be non-invasive. There is no surgical procedure needed. We can just record the electrical activity at the body surface. And the second, the electrical activity can affect the brain, transfer to the brain directly. So the brain afferent terminals on the stomach is the electrical activity. It's highly overlapped with the ICC neurons that I mentioned previously. So it's highly possible that it can receive the direct electrical activity and send that to the brain. So if we recall the electrical activity from the body surface, does it really reflect the gastric activity? The answer is yes. So there is a mathematical model supporting this idea. So they build a model that from the single ICC cell, for each ICC cell, there is an action potential generated. It appears periodically, and those cells group into ICC clusters and generate the local field potential. And later on, the activity propagates to the whole stomach and generates an organ level propagation of the electrical activity. In the end, this organ level electrical activity can propagate to the body surface and can be captured by us. So in our study, we try to capture the body surface far field potential, which reflects the gastric activity, which is so-called gastroelectrogram and short for EGG. And the way we do that is that we create a patch electrode which include a lot of channels. And this patch electrode can be attached to the belly of the rat. And we connect this electrode to an amplifier that records the far field potential. Then we want to know whether the electrical activity we record is really reflecting the gastric function, which is a contraction. So we did an experiment. We record the EGG signal inside the MRI, and we also perform the fast imaging of the stomach. And using imaging, we can capture how the stomach contraction moves, how the gastric stomach contracts. So we extract the contraction wave from the one part of the stomach, which is the antrum, and try to correlate the stomach contraction with the gastroelectrogram, which is EEG we recorded. And there is a high synchronization between the EGG and the gastric contraction, which prove that the EGG signal we recorded can be used to representing the stomach contraction. Next, how should we get the brain signal? So the way we do it is that using the functional imaging to get the brain signal. So the functional imaging is capture the signal which are called blood oxygen level dependent activity. So it's not it's not a direct recording of the neural activity. Instead, it's used some mechanism behind some relationship between the neural activity and the blood flow. So when the brain is active in normal stage, the blood flows through the regions, including two parts. The blood flow from through the brain, including two parts. Which one is the oxyhemoglobin, which carries the oxygen, and another is the oxyhemoglobin that does not carry the oxygen. So the oxyhemoglobin flow into the tissue and carry the oxygen to the tissue, and the tissue use up the oxygen and change the oxyhemoglobin into the deoxyhemoglobin. So when there is a brain activity, there's neurons depolarize. So there is oxygen consumption increase, and it decrease the local oxyhemoglobin. And because of the decrease, there's more blood flow into the brain region and try to bring more oxyhemoglobin into this region to support the consumption needs. So there is so the local brain activity end up with an increase in the oxyhemoglobin, and there is a property of the hemoglobin. So there is ions in the oxyhemoglobin. So when the hemoglobin is oxygenated, so the ion is not paramagnetic. But when it becomes the deoxyhemoglobin, the ion become paramagnetic. And the paramagnetic ion can affect the local field, and cause the susceptibility artifact, and which decrease the MRI intensity. And the the more oxalic hemoglobin flow into the brain region, which will end up with the increase of the bold activity. So using bold signal, we can reflect how the local neuron activity works. So the reason why we did not directly record the neural activity but record the bold activity is that using the fMRI image, we can cover the whole brain at in just 1 second or less. But if we record the neural activity can only be recorded as a local field. So it can only reflect a small region in the brain. And if we want to cover the whole brain, we either lose the spatial resolution or we cannot include every detail in the brain. So that's why we use the record the bold activity at the first stage. So now we have the gastric electrical activity from EGG, and we have the brain activity from MRI. And the next step is that we record the EGG during the MRI, perform the simultaneous recording, and record the EGG inside the MRI and during the imaging acquisition, and can get the gastric activity and the brain activity. Next, we want to compare these two signals. So here is just the illustration of how the gastric activity and the bold activity looks like. So the gastric activity is highly sinusoidal. So each peak indicates one contraction of the antrum. And the cycles of the peak is relatively consistent, which end up with one dominant frequency of the gastric activity. And for the brain, it's a little bit complicated. The frequency component of the bold activity is really complex. It can vary from the 0.1 hertz to right now we think that the bold activity can up to the 0.2 hertz. So there is a lot of frequency component inside the bold activity. How should we compare these two activities? So the way that we use is a cross-spectrum analysis. We try to find the coherence at every single frequency band. And we only interested in the coherence at the frequency, which is the dominant frequency of the gastric activity. And by test the consistency of the coherence at this frequency across animals and across trials, we can uncover the gastric network, which is shown here. So there is a lot of regions are included in the gastric network, not only include the somatosensory cortex, motor cortex, visual cortex, and cerebellum, which is consistent with the human results. It also includes some regions such as the auditory cortex, prefrontal cortex, and we also found some thalamic and brainstem nucleus in the network. So far, we know that there is a stomach-brain synchronization, which means that the brain is always listening to the stomach. It's listening to every contraction coming from the stomach. So since the brain is listening to the stomach, is there any phase delay between the two organs? So to test that, we summarize the phase across the brain as shown in this map. So as you can see that there is a phase difference between the two signals, and the phase difference can change across the brain. To take a close look at the phase difference, we average the fMRI signal and the EGG signal and compare these two signals directly. And it can be seen clearly that for the brainstem nucleus and the somatosensory system and the motor cortex, usually the phase delay is relatively small. It suggests that these brain regions have a closely look into the stomach activity. However, for the visual cortex, auditory cortex, and some prefrontal and amygdala, the phase delay between the two signals is in general large. So it may suggest that the stomach can affect the brain activity in those regions, but the effect is not very strong. Then to move forward, since we have the phase delay, we want to ask a question. Is there any propagation of the gastric activity in the small brain region? So the reason we ask this question is that for our sensory and motor system, there is a topographic organization. For example, it's a classic illustration of this topographic organization. For example, if we want to control our food, the brain regions are responsible for this contraction. It's more in the middle of the motor cortex. But if we want to control our hand, this region that responsible for this function is maybe in this region. And if we want to control our face, the region that's responsible for the function is in another place. So there is a topographic organization. So is there a topographic organization of the stomach? So the stomach is not only have one, the gastric activity in the stomach is not in one phase. Actually, it's a propagation included in the gastric activity. So there is, in general, pacemaker regions in the stomach, and they generate a periodic signal. And the periodic signal can propagate over the stomach along the longitudinal direction. And during the propagation, there is at the same time, there is a significant phase delay between the signal come from one region to another region. So the brain recognizes such kind of signal propagation in the brain to have a feeling of that problem. We try to find the phase delay in a small region, start from the brainstem nucleus to the motor cortex and the somatosensory cortex. And turns out that in the brainstem nucleus, which is the first relay between the stomach and the brain, there is a phase propagation in the small region. If you see clearly that the voxels in the lateral side has a small phase delay, but the signal in the medial side has a longer phase delay, and similar in another region of the brainstem nucleus. And it's a little bit different in the somatosensory region and the motor cortex. If we check the phase delay, phase propagation in those regions, there seems like there is a clear cutoff in the regions, and one part of the motor cortex has a shorter phase delay, and one part of the motor cortex has a longer phase delay, similar to the somatosensory cortex. It's just to give us a sense that maybe there is also a topographic mapping of the stomach activity in the brain, but further study is needed to better illustrate how the topographic organization of the stomach in the brain. So far, we validated that there is a stomach-brain synchronization, and we can use the phase information to tell how the stomach activity propagates in the brain. So the next question we want to ask is, what's the origin of the stomach synchronization? Because the fMRI is not a direct measurement of the neural activity. It can be affected by the blood flow, or it can be affected by some other physiological noise. So we want to ask whether it's neuronal. If it's neuronal, which nerve connection is responsible for the synchronization? So we hypothesize that it's neuronal. It's because that the stomach and the brain are highly connected by the nerve. So there is two major parts of the nerve connection. One is the sympathetic pathway, and another is the parasympathetic pathway, which is the vagus nerve. And in these two different pathways, the vagus nerve connection is thought to be the most important connection between the stomach and the brain. And there is a well-established vagal stomach connectome in rats, which maps how the afferent vagus nerve ending present in the stomach. And it's clearly see that the vagal afferent terminal can cover the anterior stomach wall, and more importantly, the nerve ending often overlapped with the gastric neurons, which is the ICC neurons I mentioned previously. So in this map, the ICC neurons is in the blue color. It stands in the blue color, and this stripe-like shape is the vagus nerve afferent ending. So it's the vagus nerve afferent ending is often overlapped with these ICC neurons, and they often have the synaptic connection directly with the ICC neuron. It suggests that the ICC electrical activity may directly transmit to the vagus nerve and goes to the brain. So we hypothesize that the vagus nerve is the major contribution to the stomach-brain interaction. So to test that hypothesis, we did a vagectomy on rats, which means that we cut the vagus nerve at the cervical level, and after the surgery, there is no vagal connection between the stomach and the brain. And after the surgery, we recall the EGG activity together with the functional MRI and try to perform the coherence analysis as I described previously. And it turns out that after the coherence analysis, after the vagectomy, there is no significant gastric network can be found in the brain, and which is shown here. As a comparison, this is the gastric network that was in the control group. So the vagectomy significantly desynchronized the stomach-brain synchronization. To further validate this hypothesis, we performed the two-way t-test to compare the coherence between the vagectomy group and the control group. And indeed, the coherence at the vagectomy group is significantly lower than the control group. And the difference is more significant in the region of the somatosensory region, striatum, and the vagal and the thalamic nucleus. So far, we proved that we validated the synchronization between the stomach and the brain, and proved that the vagus nerve mediated this synchronization. So the next thing we want to do is that, is there another format of the stomach-brain synchronization? So the stomach is contracting. It's a periodic signal. It's a periodic thing. It's contract over time with relative like a sinusoidal shape. However, the periodic is not the only feature of the stomach contraction. There is other feature. For example, the stomach can contract stronger or weaker. We and our collaborators did an experiment that put a strain gauge, implant a strain gauge on the stomach surface. So it's a picture of the during the surgery. So this whole thing is the stomach, and here is the full stomach, corpus, antrum, and we put a strain gauge sensor on the surface of antrum and let the animal recover. So we can record the strain change in the stomach, and the animal at the same time the animal is free behaving. They can eat, they can drink, they can sleep, they can be active. So we record the strains of the stomach over time, and we can clearly see that each peak indicates one contraction of the antrum, and if there is no peak or the peak is smaller, it indicates that the contraction is weaker or there is no contraction. So it can clearly see that the contraction strength is not consistent over time. Instead, sometimes it's stronger, sometimes it's weaker, and sometimes it's a little bit silent. So the contraction strength is also an important feature for the stomach function. So how do we access this contraction? So since we have the electrical activity, can we use the electrical activity to reveal the strain change in the stomach? The answer is yes. So there is an old paper support this idea. They record the electrical activity from a local ICC neuron group, and they also record the strain gauge signal from the stomach. Together with this invasive signal, they also record the body surface signal, which is the EGG electrogastrogram, which is showing in this one. So when the contraction is stronger, and sometimes the contraction is weaker. So when the contraction is stronger, the EGG signal, the amplitude of the EGG signal also increase. And when there is no contraction, the amplitude of the EGG signal decrease. So we can estimate the stomach contraction strength by evaluating the amplitude of the EGG signal. So that's what we do. So here we have the far field EGG recording, and we calculate the power of the EGG recording. And in our recording, we can clearly see that the amplitude of the EGG signal is fluctuating over time. Sometimes it's stronger, sometimes it's weaker, and sometimes it's super strong. So now we use our EGG signal to find a feature of the EGG signal which can reflect the gastric contraction strength. The next step is how should we find the relationship between the contraction strength and the bolt activity? So because the bolt activity is super complex, it's hard to reveal the relationship between the bolt activity and the EGG power. So the way we do that is that we assume that there is a black box between the EGG power and the bolt FMR signal. And the black box can be described using an H function. And by convoluting the EGG power, which is the X, with the H function, or we can plus the noise, we can get the bolt activity. That's the assumption. So, but what is the H function? We don't know that. So the first step we do is trying to solve this H function. And then after getting this H function in a different data set, we can convolute this H function with the EGG power and calculate the estimated bolt activity. By correlate this estimated bolt activity with the real bolt activity, we can know that whether the brain is really listening to the contraction strength changes. So the answer is yes. And there is a few regions that are listening to the change fluctuation of the contraction strength. The first region is the NTS neuron, NTS, which is the first relay between the stomach and the brain. And another few regions are including the thalamic nucleus, and most importantly, the somatosensory region and the striatum, and the insula. So the somatosensory regions can help us, may indicate that our sensory system is always listening to the contraction strength. And the striatum is responsible for the food reward. For example, if we all like candy is sweet, so we want more candy. Or we did a thing when we are a child. We did some good things, and our parents give us prize, which is candy. So we want to do more good things to get the candy. So that's what the function of the striatum. So this part also listening to the contraction strength. Another part is the insula. So by our understanding, insula is a high representation of the visceral sensation. So it's listening to the visceral organ that responds correspondingly. So all those regions are all listening to stomach contraction. But how those regions respond to the gastric contraction? So for the regions like the brainstem nucleus and the somatosensory regions, they respond to the contraction changes quite quickly. So whenever there is a contraction change, we can see that the NTS nucleus and the NTS and the somatosensory neurons quickly respond to the contraction strength, the change of the contraction strength. But for some high-level areas, for example, in insula, the response is quite slow. So the change of the contraction strength can have a longer effect on the neural activity in the insula. So instead of contraction strength, is there any other features of the stomach? Yes, there is. So the stomach contraction is not always stationary. It can change over time. Sometimes the stomach contraction can be slower, and sometimes it can be the electrical activity in the stomach can be super fast. So when the electrical activity is super fast, it's hard to engage the contraction of the stomach. So they are all features of the stomach activity. So can we take that features out from the EGG recording? Yes, the answer is yes. So this is the illustration of the EGG activity that's similar with our far field recording. In the normal stage, there is we can clearly see that each peak of the gastric activity is slower. In human, it's around 3 peaks per minute. But sometimes the fluctuation can be faster, which is so-called tachygastria. And in those stages, the electrical activity has the cycles of that activity is faster than normal stage. So in human, usually it can reach to 5 or 6 cycles per minute. In another stage is that when the stomach contracts slower. So in those stage, we can just find 1 cycle of the electrical activity in a single minute. So yes, so how can we find out that in our data? Yes, we did the power spectrum analysis across time. So the color indicates the power at a certain frequency, and the x-axis is the time, and the y-axis is indicate different frequency components, which is how many cycles per second. And in red we define the normal gastric frequency is around 4 to 6 cycles per minute. And if it's higher than 10 cycles per minute, we think it's tachygastria. And if it's lower than 3 cycles per minute, we define it as bradygastria. And then by doing this power spectral analysis, we can find when the tachygastria occurs during the time and when the bradygastria occur during the time. And we use a similar way as I described previously to uncover the relationship between the fluctuation of tachygastria and the brain activity by resolving the H function and correlate the estimated both activity with the real activity. So here is the result. So there is still have brain regions that respond to the tachygastria or the bradygastria. The region may different, but the region is different. The region that responds to the 2 different stages are different. But it's usually covers the brainstem nucleus and somatosensory regions and motor cortex. And especially for the bradygastria, the amygdala also responds to that. It may suggest that the occurrence of the bradygastria may affect our emotion, how our feel, whether we are happy or we are angry. To wrap up, so we find that the brain is synchronized with gastric activity with different phase. And the second thing is that we know that the brain is monitoring the stomach contraction strength over time. And not only that, the brain also alert to all the dysrhythmia of the gastric, for example, the tachygastria and the bradygastria. So to move forward, what's the future direction? So as mentioned previously, we would like to uncover the topographic organization of the stomach in the brain. So why is it the topographic organization is important? Because the well-organized phase difference on the stomach can result in a coherent, result of a synchronization, synchronized pattern of the stomach contraction. So here, this figure illustrates the phase of the phase of the contraction on the stomach at different regions. So in the healthy subject, the contraction has well-defined patterns along the stomach. So there is a region that has a higher contract, and there is a region that is right now relaxing. But in the patient, for example, the gastroparesis patient, their stomach contraction pattern is totally disrupted. So the reason that by looking at the topographic mapping of the electrical activity in the brain, we can know that we can we can try to figure out that in the patient with gastroparesis with a disrupted stomach contraction pattern, we know how the brain responds to that, and by looking at that, we can identify help with the diagnosis process. And another future direction is that they can help with the diagnosis process as I described previously. There is the autonomic neuronal disorder can result into the gastroparesis, and it can come from the damage of the vagus nerve. It can also come from the neuron disorder, for example, the Parkinson's disease, in the epilepsy or other autonomic disorders. So by looking at the stomach-brain interaction, we have the potential to find out which part of the neuronal dysfunction causes the gastroparesis, which can help with the diagnosis process in clinic. And the last thing is that by knowing the stomach-brain interaction, it may help with the therapeutic process. We already know that there is an electrical solution for the gastroparesis, but the mechanism behind it is not very clear. So for the electrical stimulation, at the beginning, people think that the electrical stimulation can work as a pacemaker and can help to pace the stomach contraction. But this pacemaker only works well for the diabetic-induced gastroparesis. Why it doesn't work for others? So now there is some recent thoughts on the mechanism of the electrical activity. They think that the electrical activity not only paces the stomach, it also affects how the brain responds to that. But we don't know how the brain responds to the stimulation and what kind of stimulation is good for the brain input and what stimulation is not good for the brain input. By studying the stomach-brain interaction, we can help to solve this problem in the future and help to fine-tune the stimulation parameter and method pattern to help with the stimulation therapy. So in the end, I would like to thank for my lab mates who help with the research idea, help with the analysis, and help with the experiment. And I would also like to thank the funding source from the SPARK program and the people from the SPARK team that support me for this study. Okay, thank you.
Excellent. Thank you, Cherry. We will take any questions now. Does anyone have any questions for Cherry about her presentation? I actually had a more general question. So you talk about the relation between the brain activities and the stomach system. Do you see it as a cause-effect relationship where one is definitively a cause and one an effect, or is it more complex and interconnected than that?
That's a great question. So, from our data, we don't know that, but there is some general thinking about the causal relationship. So in the stomach, the ICC neuron generates a periodic signal, so which is a source that we know. So it's highly possible that the brain receives this synchronization instead of sending this synchronized interaction. However, the brain can affect how the stomach contract, can affect the frequency and the period of the. So maybe in the brain, in some regions, it just receives the synchronization signal, and some brain regions can process and send the signal back to the stomach. So it's a little bit complex for the causal relationship between the two. And we would like to try to find out some way to differentiate, to try to find out the causal relationship, which is really helpful and essential for the future, especially for us to fine-tune the stimulation parameter in the future. Yeah, that's a great question.
Thank you. Any other questions for Cherry? If we don't have any more questions, I would like to say thank you to Cherry for preparing and giving your presentation. It was excellent. Um, Thank you to everyone who came and was an audience for her. If you have any feedback, please go to our website and provide both our speaker and us feedback of how we can make these presentations better. And then one last reminder, if you didn't get a chance to, fill out our attendance form so that we can see how many people are coming and gauge interest on our talks. Thanks again to everyone who came, and we look forward to seeing you guys next week. Thank you.
Thank you.